Nice.
Hello everybody, my name is Jonathan Ellis. I also work at the University of Plymouth. Like Mary, I spent the first part of my career studying bumblebees and working on conservation genetics. I've worked on other animals as well, like salmonid fish, and more recently on fungi, but I've also recently got into doing work on honeybees. So I'm going to talk to you about introgression and loss of locally adapted traits in honeybees. What I'll do first is I'll just give people the fundamentals about introgression, how we measure it, what it is, how it breaks down local adaptation, and then I'll raise some points for discussion, and then I'll hand over to Matthew, who'll talk about the state of introgression in the UK. Just a reminder about some fundamentals of biology. Obviously, in our cells, we have nuclei that contain the chromosomes and nuclear DNA, but also in our cells there are lots of organelles, including mitochondria. The hackneyed phrase about mitochondria is that they're the powerhouses and batteries of our cells. And they're interesting little things. They're really important, actually, for things like healthy aging and avoiding metabolic diseases. But another important an interesting thing about mitochondria is that they contain their own genome. So in our cells, we have the genome in the nuclei, actually in the chromosomes, and then we've got mitochondrial genome as well. And that's important for how we measure introgression in bees. So introgression is sort of a mixing of genetic material from one lineage into the background of another, and it results in admixture, a mixing of the genetic material from both sorts of lines. So on my slide here, imagine that we've got different lineages of bees, an M lineage and a C lineage, and we've got males and females of both. And when we cross from, it says B to A, should say C to M, we end up with a mixing of the genetic material between the two different lines. So let's say a female C lineage bee mated with a male M lineage bee, we'd have offspring that would contain the C lineage mitochondrial DNA, and that happens because mitochondrial DNA tends to be maternally inherited in most animals, but you get a mixing of the nuclear DNA of the M and C lineage bees. If it were the other way around and we had a female M lineage bee mated with a male C lineage bee, the offspring would have M lineage mitochondrial DNA, and then of course a mixing of the M and C nuclear DNA. So when we've got a population of bees that have been interbreeding between different lines, we see, we can detect both M and C lineage mitochondrial DNA in the population and look at the relative proportion of M and C lineage mitochondrial haplotypes or DNA sequences, or we can use nuclear markers and we can detect the mixing of the genetic material from the nuclear DNA in the two, in the resultant hybrid offspring. So, in a nutshell, introgression is the mixing of genetic material from one line into the background of another. Now, that can be a problem because this process of mixing the genetic material between different lineages breaks down local adaptation. And there's another hackneyed phrase that scientists use about local adaptation and how it arises, and they say it's the result of co-adapted gene complexes, which sounds more complicated than it really is, like lots of terms in genetics. And it's just the assembly of different genetic variants at different regions in the genome. So imagine we had three genes, gene A, gene B, gene C. These occur at different positions in the genome of B. And over time, natural selection assembles particular genetic variants of these regions. And they could give be an advantage. For example, it might favor a combination of genetic variants for several reasons which underlie resistance to a local parasite strain, for example. The problem is when you get the introgression and the admixture occurring, that this is then broken down so that you get a mixing of the genetic variants. So we get new genetic variants being introduced into the population, and that disrupts these locally adapted gene complexes that have been assembled by natural selection over a long period of time. But rather than maybe thinking about the sort of background genetics, it's always helpful to have a concrete example. And this is very similar to something in conservation biology called outbreeding depression, where people move individuals from one population into another population, hoping that that will give a rare and declining population a boost. But sometimes it sort of backfires and there are negative effects of that. And a famous example is with Tatra mountain ibex, where individuals from the Middle East were moved into the northern part of the range, and the hybrid offspring of the Middle Eastern ibex and the ones in the Tatra, they mated later in the year and they ended up calving in the winter. So, and there was a massively reduced survival of the hybrid offspring. So that's this sort of outbreeding depression and loss of local adaptation occurs as a consequence of the mixing of the genetic material that breaks down this local adaptation that natural selection has assembled. But the question is, like, how can we measure that in populations? And we can use various sorts of genetic data to do it. Here on the slide, got an image of some DNA sequence data. Each row is a different individual, and each column is a particular homologous base in the DNA sequence. And if you look through that, you can see all the sequences are identical, apart from at one location where we've got this C and a T here. And this is what you call a single nucleotide polymorphism. And there are lots of different ways that we can measure genetic variation in the population, but with the advent of genome sequencing and more advanced technologies, these days people routinely analyze what single nucleotide polymorphism data. So we can use this to investigate the admixture and the mixing of lineages. Because imagine we've got 3 different genetic clusters in a population. I've got them up here, 1, 2, and 3. And these are different, say, populations of the species, and they've got a different genetic signal associated with the different populations. And we might have different single nucleotide polymorphisms associated with the different population clusters. So associated with this cluster, we might have, say, an A in the first position, a T, C, and C. This isn't a sequence, but different regions across the genome where there are different SNPs, different single nucleotide polymorphisms. In the second one, we might have a T, an A, a G, and a G, and then the third one, a C, T, an A, and a G. But the question is, when we've got an unknown individual then with a genotype like this one here, how can we assess its ancestry? Because it's got a T that suggests it's from the second population. If it had a T in the second one, it could be from the third or the first population. And A here suggests it's from the last one. A G here could be from either one of these two. You can see it rapidly would get quite complicated to try and assign the ancestry of an individual. But thankfully, statisticians have invented lots of complex packages that we can input reference DNA data from clusters and test individuals and work out their degree of admixture and use that to understand the ancestry of the different individuals. So in practice, what we do is we genotype bees, we ask software package to put those bees into clusters for us, and then we can look at the ancestry and the genetic makeup of the different individual bees based on the sort of genetic signal that comes from the genetic data. One thing it gives us is a value that's called a Q value, and this goes like 0 to 1. And 1 would be a completely pure bee, and when you get admixture and mixing of the populations, it would come down and decline to, say, if it was 2 bees that recently just hybridized, it would be about 0.5. So we're interested in this question here. What proportion of the genotype of an individual is attributable to a particular reference population? We use these Q values that are also known as admixture coefficients to tell us about the degree of introgression and mixing of the populations. And often you might see, if you've been to these sorts of talks before, a figure that would look like this, where it's hard to see, but it's composed of lots of individual lines, vertical lines, all stacked next to one another across the figure. And we've got some reference populations here in blue and red and green, and then we've got some test population at the end where we want to look at the degree of mixing of the genetic lineages. And we can see we've got individuals here where there's some signal from a green genetic cluster and some signal from a red genetic cluster. So this line where you see a mixing of green and red is an admixed hybridized individual, whereas when you've got a line that's all one color, it's a relatively pure bee. So we get genetic information from reference populations. You can get genetic information from the test populations. You can put these data into software packages and it spits out these values called Q values, and then you can inspect the Q values to see how introgressed particular bees are. But I think it's important to raise some discussion points about these sorts of measures that scientists use. So the values that you get from this kind of analysis depend on the number of reference populations that you use. So you ask the software to tell you about the number of clusters in the data, and you pick a suitable number of clusters, or you can do that based on your biological knowledge of the population. So in bees, if we were interested in honeybees and the M and the C lineage, we might assign the bees to 2 clusters, M and C lineage bees, but you could also use more clusters than that if you wanted to, depending on the natural population structure of the species. That will affect the Q value. It might also depend on the genetic markers that you use. That means just the sort of gene regions that we use to get genetic information. Lots of different types that can be used. But what I really want to sort of ask today and get is, if you had your bees genotypes and you got a value back, you'll get this Q value that tells you sort of how pure your bees are. And often you might get a value like 99%, 95%, 90%. So you can look at the overall extent of the admixture in the population. But I think it's important also to reflect on, it's going to depend on where in the genome of a bee those admixed variants occur, how it interrupts the local adaptation. Because if you disrupted those gene complexes that I mentioned earlier, then you could have a low level of admixture in the sort of wrong places in the genome that then would affect the local adaptation of the bee. Or you could have quite a lot of admixture, but perhaps a bit in other places in the genome there wouldn't be this loss of local adaptation. And I think an important question that really needs to be addressed by scientists is the link between the genotypic data from bees and the colony level phenotypes, and trying to understand more about what level of introgression and admixture breaks down that local adaptation. And then we can have a better informed idea of what a suitable threshold value is for the level of admixture in the population. I think that would be plug an interesting gap in the data. But there are also some other interesting things about these sorts of processes. So even in the face of strong gene flow or strong rates of imports, if selection is strong enough on the locally adapted variants, you can maintain locally adapted bees in the population by natural selection. There's this balance between selection and the flow of genetic material into the population, and it depends on the balance of the selection coefficients that maintain the genetic variants and the rate and extent of gene flow coming into the population. But that means even in the face of strong imports, you can still see, relatively pure bees maintained in the population. And that might be something that you see in the country's data. Some people who are less concerned maybe about imports might say, admixture increases the genetic variance of the population, and that's important from a breeding perspective. Generally, having high genetic variance is sort of good for people who want to select traits in a population. But the thing is that once you've introduced new variants into the population, then they're sort of there for a really long time, and it breaks down the natural population structure of the species that's evolved over really long periods of time, and it can quite easily break down these locally adapted populations, and then we lose the sort of races and varieties of bees that have emerged naturally over a long period of time and are better adapted to the local conditions. And evidence shows that local bees sort of perform better than imported bees in the population. But I just thought I'd mention that. And another really interesting thing about this sort of process is that some of the variants that are introgressed into the population can be adapted and give individuals a selective advantage, and then they might be maintained in the population for a long time. There is this other process called adaptive introgression. That's actually interesting in humans. I investigated my own ancestry in the past. I've sold all of my genetic data, rather foolishly, to an American company, but they told me that I have more Neanderthal DNA than 78% of customers that have done this. So I guess that makes sense, because the other thing it revealed about my own ancestry is that I'm really broadly Northwestern European, like mostly British and a little bit of French, German ancestry in my own lineage. But that's happened because early humans and Neanderthals mated with one another, and then over time some of those Neanderthal variants have been maintained in the population. They could be maintained by selection, but I think that's also just shows that once this introgression has occurred, how long these variants can persist in a population for a long period of time. And that's what I wanted to say, just to give an overview. I think my job really is to sort of give an overview of these measures. Hopefully that was clear enough. I know we've got people from probably a wide range of backgrounds in the audience. Some people are quite familiar with genetics. Some people, trying to bring back memories from high school and things like that. But in a nutshell, we've got these Q values that measure the degree of admixture. And the closer it is to 1, the purer a bee is. But we need, I think, to know more about what level of admixture would begin to break down these local adaptive variants. But you can see that introgress variants can then persist in the population for a long time. So that's me done, and now I'll hand over to Mark, who will talk more about those measures in the UK population. Oh, sorry, that's Matthew. I thought it was changed. Sorry, I thought it was going to be Mark, Matthew's colleague, but it's Matthew today. So.